Our speaker today is Sam. Hello, Sam.
Hello.
So Professor Fountain is currently at the University of East Anglia in Norwich, chair of pharmacology, and also the associate pro vice chancellor. Excellent, right. So you actually started off old school, so working with David Beach in University of Leeds, maybe on piezo, maybe not, I'm not sure, I haven't checked. And then you moved to Manchester to work with the great Alan North, and I think that's probably where your interest in P2X receptors started. So there's been a lot of interest in P2X, as Stan mentioned in his abstract, mostly driven by recent clinical work on P2X2/3 antagonists for chronic cough, and a couple of those have been approved now for the market. And Sam actually in his lab did some work on Merck's Gefapixant. But he's not going to talk about that today. Today's going to talk about another P2X receptor, which has been long of interest in drug discovery, and these are P2X4 receptors, which were perhaps best known for their role in pain, potential role in pain. But Sam's also going to talk about their potential role in vascular secretion and pulmonary. So over to you, Sam.
Great. Thank you so much, Mark, and thank you very much, Ali, for the invitation to come and talk today. So as Mark said, I'm a professor of pharmacology at the University of East Anglia, which is based in Norwich. If you don't know where that is, it's a medieval city just north of Cambridge in the county of Norfolk, which boasts some of the best coastline in the United Kingdom. If this interests you, bear that in mind when I'll introduce you to some opportunities at the end of the talk, but it really has some exceptional coastlines. A brief overview of what my laboratory is about. Essentially, we're interested in some of the main cardiovascular and metabolic respiratory tissues, so airways. Really interested in the role of ATP, extracellular ATP, as a neurotransmitter, but also in its role being secreted by other cell types. So the family of receptors I'm going to focus on today, according to Artem's analysis, is the least interesting receptors. Is that right? That's what I got for, or the least studied, I should say. So hopefully I'm going to change that today. It's probably a good thing for me and my group that they are the least studied. But we're going to look at the channels that are activated by extracellular ATP, and they're called P2X receptors. And P2X receptors are a family of non-selective cation channels. So when they're activated, they depolarize cells, they elevate intracellular calcium. And when we look at the structure of P2X receptors, they're rather unusual when we look at the other types of neurotransmitter receptors, such as the glutamate receptors and the cis-loop receptors, that P2X receptors have this double transmembrane topology, which is unusual for ion channels, but also that they form functional trimers. So the active channel is trimeric. And a lot of work recently has gone into solving structures of P2X receptors. We now have some very, very good structures of P2X receptors with ATP bound, also more recently with some antagonists bound, which is giving us a lot of useful structural information. And the important thing to think about P2X receptors is that when they bind ATP, they don't hydrolyze it. They're not pumps. It's just a ligand-protein interaction that is reversible. And it's the ATP that binds in an subunit binding interface on the outside of the receptor, and it takes at least 2 molecules of ATP to gate the channel. Irritatingly, when we look at P2X receptor families in mammals, of which there are 7 receptors, these allosteric sites for ATP binding are highly conserved structurally and primary sequence-wise, making them incredibly difficult to drug to give selective blockade or enhancement of P2X receptor subunits. So there's a big drive at the moment, including some of the work that we'll talk about looking at allosteric modulators and which areas of the receptor modulate allosterism. P2X receptors have been known about for quite a number of years, and AstraZeneca did an early trial looking at P2X7 and rheumatoid arthritis that didn't give any significant outcome from that trial. So a lot of interest was lost around P2X receptors and their potential as drug targets. But we worked in this area with of chronic cough with Merck. And chronic cough is interesting because in adults, it's something that's characterized of coughing for more than 8 weeks, 10% of respiratory referrals to secondary care, and about 2 to 18% globally, there's a prevalence of chronic coughing. And people are refractory to common treatments. And there's a big drive also to look for non-opioid based treatments for chronic cough. Many of you might be aware that cough is treated by subanalgesic codeine, for example. So we got very interested in one of these receptors called P2X3. And the reason it's interesting in terms of cough is that P2X3 is, when you look throughout the body, is almost exclusively found expressed within sensory afferents. And that's true for the neural networks that regulate the cough reflex in humans. So you can see up here in these nodose and jugular ganglions are where you find the cell bodies of these sensory afferents that are innervating the airway and pulmonary lining. And if you look, do immunocytochemistry, you can see that these sensory neuron cell bodies express large amounts of P2X3. So we collaborated with Merck in the preclinical development of a drug called Gefapixant, which was named after Jeffrey Bernstock, who was the pioneer of the area of purinergic signaling, to be involved in the preclinical development of some nanomolar P2X3 antagonists, and this work is published here. Importantly, Merck and its predecessor, Afferent Pharmaceuticals, led by Anthony Ford, undertook a number of clinical trials to show that P2X3 antagonists are highly efficacious at reducing cough frequency in individuals. And the reason it works, we believe, or we're not certain, is that chronic cough, just like bladder hypersensitivity, is believed to be a result of hyperreflexia. Some increased activity of that afferent communication from these tissues drives up reflexes such as bladder hypersensitivity and chronic cough. So if you can dampen the communication of those sensory neurons, you can reverse these types of disease and disorder. Gefapixant has recently been approved as a new medicine for the treatment of chronic cough, and importantly, it's not an opioid. So this is the first drug that's been approved that targets P2X receptors and has led to a resurgent interest in P2X receptors as drug targets. I'm going to look at a different receptor today, which is the P2X4 receptor, and look about strategies that might involve its antagonism, but also its potentiation. And this is something we're particularly interested in cardiovascular biology. So if we look at the sort of diseases that P2X4 is involved with and that you might want to potentiate, such as alcohol use disorder, hypertension, ischemia, multiple sclerosis, and anxiety, but also some conditions where you might want to block P2X4, and many of these are around neural damage and pain and inflammatory mechanisms. We're interested in looking for allosteric sites within the P2X4 receptor that we can modulate both to enhance and inhibit the activity of the receptor for different disorders. So there's not really a lot of information known about allosteric sites within P2X receptors. So we started to explore this by looking at some known commercially available antagonists. And here's an example of one called 5-BDBD. In the past literature, it's not particularly well characterized, and there was some lack of clarity whether it acted competitively or non-competitively, but we show that it actually works as a non-competitive antagonist. So it's quite good, blocks P2X4 with relatively good potency, but important for the study I'm going to talk about, it doesn't block P2X2 at all, and it works by blocking the response maxima rather than shifting the ATP EC50. So it's a non-competitive antagonist, suggesting that it might work by an allosteric site, something away from the orthosteric site for ATP binding. So we use this information and this difference in sensitivity between P2X4 and P2X2 to do some chimeric channel generation where we systematically replaced parts of P2X4 with parts of the insensitive P2X2 channel and looked for dropout insensitivity for 5-BDBD, identifying areas that are important for conveying the effects of 5-BDBD. And you can see that one very nicely in red pops out of this chimeric study. Most of the other chimeras, you find that there's conservation of 5-BDBD activity, but not in this region. And when we look at where that region is shown in red, it's away. So this is an ATP bound in its pocket here. It's away from the ATP binding site. And this is a top view. We can find these sites kind of sitting just around the top ends of the receptor here. We use this information to generate some predictions about where the molecule binds, and that's shown here. And what we identified is that this molecule binds to an allosteric site that is formed at the interface of two subunits. So that's quite interesting, because there's a big interest in P2X receptor heteromers, and potentially this is a route for drugging heteromers over homomers. So we tested these binding hypotheses using some molecular dynamic simulations. So I'm just giving an example of this here, where we're able to do a simulation, for example, for this carbonyl group and look at its proximity to these arginine residues, either R82 or R301. And you can see that the 301 interaction is stable, and that this line going wiggly and moving away means that that is an unstable interaction. So it favors this model where the carbonyl group of the 5-BDBD molecule is interacting with this arginine 301. We can also take another approach when we look at the amino acids that we find within that allosteric domain and make them systematically more P2X2-like and drop out 5-BDBD activity. And you can see this for a number of channels. Here's the alignment here, particularly around 301. You can see the difference between the P2X receptors. And as we systematically make it more P2X2-like, we find some key residues within our predicted allosteric site that mean the receptor loses sensitivity to 5-BDBD. So this mutagenesis supports these in silico predictions. We also looked at it the other way around to do some medicinal chemistry by changing functional groups within the antagonist molecule that we would predict would be helpful or would hinder the interaction if our binding prediction was correct. So we made a lot of different chemical analogues of 5-BDD. I don't have time to go through all of this information today, but the outcome is that any introductions that we put on the molecule that we would predict from our in silico models to hinder binding, we see dropout of activity. So together, that molecular work and the chemical work and the in silico work have helped us identify a novel allosteric site in the human P2X4 receptor. Interestingly as well, when we looked in our molecular models and we looked at the allosteric site, when we looked between models of the open and closed state receptor, we saw quite a significant conformational change within that allosteric pocket. So here is the allosteric pocket, such that when the channel opens, that pocket becomes constricted. So the space becomes a lot tighter. And we might predict in that scenario that 5-BDBD, if this were the allosteric site, would have a harder time blocking the channel. So we tested this using some nanion equipment, which was particularly good because we needed some very rapid solution switching to do these types of experiments. So in this first panel, what I'm showing is the currents before and after if we've pre-applied 5-BDBD. So if we put 5-BDBD onto the cells, these are stable cells expressing human P2X4, you can see that it blocks the channel nicely. So that would infer to us that if you add the drug to the closed channel, you can block. How can we look at its effect of applying in the open state? So in these experiments, what we do we apply agonist, and you can see elicitation of the current, and then we rapidly switch to ATP plus the drug we're interested in or its vehicle control. So you can see here on the red line that actually that 5-BDBD actually only has marginal inhibitory activity when we expose the drug in the open channel state. And this isn't true for some other drugs. There's another known allosteric negative modulator of P2X4 called BX430. If we do those same experiments, we can see in the open channel that we can get rapid block of the channel, suggesting that the binding of BX430 is not hindered. And actually, there's some evidence to suggest that this molecule binds to a similar pocket, but our molecular models suggest that the size of BX430 might mean it's not hindered binding into that allosteric pocket. I want to just switch focus for the last part and talk about ways in which we can try and enhance the activity of P2X4. So a project trying to look at positive allosteric modulation of P2X4, and why might we want to do that? We're very interested in the role of P2X4 in vascular endothelium. There's lots of evidence produced by us and others to show that P2X4 is a critical ion channel in shear stress responses to blood flow in the vasculature. There's also some good indicators in the literature that suggests that P2X4 is important for regulating blood flow and blood pressure. In humans, a loss of function polymorphism leads to increased pulse pressure in human beings. There's also evidence in P2X4 knockout mice that you abolish calcium responses to shear stress, and the P2X4 knockout mouse is hypertensive and there's reduced nitric oxide nitrates found in the animal urine. The mechanism of action is that because P2X4 is a non-selective cation channel, when you get this frictional force of blood flow, this elicits local ATP release that acts in an autocrine fashion back on the channel to elevate calcium, to switch on nitric oxide synthetase, and to produce the vasorelaxant gas nitric oxide. So in this model, if you wanted to dilate a blood vessel, you would want to enhance the activity of P2X4 to drive nitric oxide production to dilate the vessel. So we've collaborated with Merck and others to look, and we're currently writing this up as a paper. We've taken the Merck compound ivermectin, which is a well-known positive allosteric modulator, and taken it to bits and made new versions to understand how ivermectin can work as a positive allosteric modulator. We've also taken parallel compound screening activities based on what we've learned from some of the chemistry here, but also from some in silico models where we've been able to identify exactly where ivermectin binds in the channel, and to produce some new drugs that are not chemically based on ivermectin that have the potential to be positive allosteric modulators. And to test their activity in the lab, we're looking at some of these positive allosteric modulators by doing pressure myography on human and mouse arteries and veins. And this is a fantastic physiological technique where you can take a vessel ex vivo from a patient or an animal, cannulate it onto these two glass cannula, and pressurize it to physiological pressure while allowing you to flow liquid through the middle of the vessel to mimic blood flow. In these experiments, and in real time, what we could measure by video edge detection is the vessel constricting and dilating. So in these experiments, what we can do, we can constrict up the vessel using something like a thromboxane A2 mimic, and then we apply this luminal flow, which mimics that flow of blood. And you can see this physiological response here, which is a flow-dependent vasodilation. So this is a way of measuring that frictional activity on the endothelium, leading to a real-time change in dilation in the blood vessel. What we're finding is some of our positive allosteric modulators can significantly enhance this flow-dependent vasodilation. And we're investigating now in the P2X4 knockout mouse whether these responses are lost in the receptor null animals. And we're also doing longer term blood pressure and cardiac rate measurements using some of these positive allosteric modulators to look for real-term changes in tissue perfusion and also mean arterial blood pressure. I'd just like to wrap up to say, obviously, I didn't do any of this work. I'm here because of all these fantastic, highly skilled people. Here's the porta patch in action, still with us. It's an excellent device, actually really useful for postdocs or students in your lab that, you know, the part of their project isn't all about electrophysiology. They can jump on the porta patch for 6 months, do their electrophysiological recordings, rather than, you know, sitting them on a stool for 6 months and making them learn manual patch clamp like I had to do. So thank you for our collaborators, particularly to AstraZeneca and Merck, who fund our work. Remember the picture of the beach. We currently have a position open for a senior electrophysiologist to come and join the group, and we also have 2 PhD studentships available, one looking at P2X receptors in vasomotor nerve activity, and the other one looking at P2X receptors in hepatic blood flow. So you'd be very welcome to apply if you'd like to. So I'll leave it there. Thank you.
Thank you very much, Sam. So we've got time for a couple of questions. Anyone in the audience here or next door?
Right, Brooke. I was wondering, because you mentioned the allosteric binding pockets, it's kind of at the interface of the subunits. Do you know whether the binding of the compound inhibits with the trimerization at all, and whether this might have an effect on what kind of the effects that you see? Could explain that as well.
Yeah, it's an interesting question. Actually, we don't really understand a lot about the mechanics of how P2X receptors assemble. The outside are disulfide bonded and interact in that way, so it might be unlikely that a ligand can separate the heteromer, possible. But it is a really important area because we actually think in real target tissues that a lot of these channels are heteromers and not homomers. And I think this is some of the problem with P2X and other knockout mice. All you can say is that part of the receptor is important. It doesn't tell you that it's a homomer, right? So we're doing a lot of work in human tissue to look in blood vessels, and there's some examples of where P2X4 comes together with other P2X receptors to mediate these sort of responses. So really important. No pointing me having a big screening campaign against P2X4 homomers if that's not the therapeutic target. But it could be interesting. I mean, there is some evidence that, particularly with P2X2 and P2X3, that the heteromultimerization can be fluid. So getting around some of the bad taste effects of Gefapixant potential. Yeah. Thank you. Good question.
All right, thanks. I think in light of the time, we should probably adjourn the first session. So I'd just like to thank all of the speakers and ask you to as well. Right, next up, we have an extended, well, not quite so extended coffee break. And I'll be first in line. Thanks very much. But